So imagine you're a firefighter,
Okay, I'm with you.
right? You pull up to a blazing house, you unroll the hose, and you just start blasting the flames with water.
Standard protocol.
Exactly. And at first it's working, the fire's dying down. But then the water you're spraying, it somehow fundamentally alters the remaining flames.
Oh, wow.
Yeah, they mutate.
Wow.
They become completely fireproof. No matter how much water you pour on, the fire just sits there, untouchable, feeding off the water itself, just waiting to spread.
That is a terrifying visual.
Right. But that paradox is exactly what oncologists are facing right now with a highly aggressive blood cancer called natural killer/T-cell lymphoma, or NKTCL for short.
Yeah, it's a brutal disease.
It really is. You hit it with heavy chemotherapy, and instead of dying, the cancer mutates into something completely invincible. But today, we are doing a deep dive into a breakthrough 2026 paper from the journal Cell Death and Disease.
And this paper is phenomenal because it finally explains exactly how these cells forge that armor.
Yes, and more importantly, how we can actually strip it away. Our mission today is to unpack all of this for you. We've got the primary research data detailing the exact mechanism, and we're going to break it all down.
We really are. It fundamentally changes our understanding of drug resistance, top to bottom. We're looking at research that doesn't just, identify a new problem. It maps out the entire molecular algorithm of how a cancer cell survives our most toxic therapies.
Specifically, a standard chemotherapy drug called adriamycin, right, or ADM.
Exactly, ADM. And we're going to trace this from the clinical outcomes all the way down to the epigenetic shifts inside the cell nucleus.
Awesome. So let's get into the mechanics of this paradox first. Normally, patients with NKTCL receive a standard chemotherapy regimen, and initially, the tumors shrink.
Right, the water works at first.
Exactly. But a massive percentage of these patients develop relapsed or refractory disease. The cancer just stops responding to the ADM.
The water stops working entirely.
Yeah. So biologically, how is a cell surviving a chemical that is literally designed to tear its DNA apart?
Well, it survives by taking a third option. So normally, a cell exposed to that level of toxicity undergoes apoptosis.
Which is like programmed cell death.
Right, right. It cleanly dismantles itself. It essentially commits cellular suicide for the greater good of the body. But under adriamycin exposure, a subset of these NKTCL cells, they don't do that. They enter a state called therapy-induced senescence, or TIS.
Yes.
They don't die, but they stop dividing. They enter this permanent, irreversible dormancy. They basically become zombie cells.
Oh, wait, I'm getting stuck here, though. Because if the cell is permanently dormant, if it's completely halted its replication cycle, isn't that a win?
You would think so.
Yeah, a sleeping cancer cell sounds entirely harmless to me.
That is the logical assumption, for sure. But it is a deadly misconception in oncology. They aren't just sleeping quietly. These senescent cells undergo massive metabolic rewiring.
Oh, really?
Yeah. To prove this, researchers exposed NKTCL cells to incredibly low, sustained doses of ADM in the lab. We're talking starting at just 50 nanograms per milliliter.
That is a tiny dose.
Yeah. Very small. But by day 30, the cultures were entirely dominated by these zombie cells, and they confirmed this using SA-beta-gal staining.
SA-beta-gal, that's a biomarker, right?
Right. It's a blue stain that highlights the lysosomal mass in aging cells. So they proved these cells were senescent. But the danger isn't that they're old or asleep. The danger is what they secrete.
Ah, okay. The SASP, right? The senescence-associated secretory phenotype.
Precisely. SASP. These dormant cells turn into biological factories. They just sit there pumping out a toxic soup of cytokines and growth factors.
Like what kind of cytokines?
Things like IL-1 beta, IL-6, TNF-alpha, and TGF-beta, really potent inflammatory signals.
So it's almost like a zombie bite.
I like that analogy.
Yeah, the dormant cell is just sitting there in a stupor, but it's drooling this highly infectious signaling cocktail into the microenvironment.
Exactly.
And if I understand the pathway correctly, when cytokines like IL-6 hit the healthy neighboring cells, it triggers their NFKB survival pathways, right?
Spot on.
So it essentially reprograms the adjacent tumor cells to take on stem cell-like properties.
That is the exact mechanism. The SASP cocktail fortifies the entire surrounding tumor architecture. It triggers neighboring cells to upregulate their own survival proteins.
It's making the whole mass stronger.
Right, making it completely resistant to the very chemotherapy that created the zombie cell in the first place.
Man, so if we're mapping this out as a clinical strategy, this completely flips the oncologist's playbook.
It really does.
We're looking at a scenario where standard chemo doesn't just fail, it actively creates these SASP spewing factories that armor the rest of the tumor. You wouldn't dare keep using the water if you knew it was doing this.
No, you wouldn't, and that raises the critical clinical question here. How do you know if a patient's tumor is going to pull this trick?
Right.
How do you identify the armor before you strike?
Okay, so let's pause here for a second and summarize this first part for you listening, because there is a lot of dense biology here.
Let's do it.
So bullet number 1. NKTCL is an aggressive lymphoma where standard chemo triggers a severe resistance mechanism.
Right, and bullet 2. Instead of dying, the drug sources cancer cells into a zombie state called therapy-induced senescence, or TIS.
And bullet 3. These senescent zombie cells secrete SASP, this toxic chemical cocktail that protects them and makes the surrounding tumor way stronger.
Perfect summary.
Which leaves a question for you to think about. Have you ever considered that a medicine designed to cure could actually trigger a biological defense mechanism that makes a disease stronger? It's wild.
It really is wild.
But okay, moving on to the next piece of the puzzle, which brings us to the proteomic manhunt. The researchers had to figure out what was chemically different about these invincible cells.
Right, they had to find the source.
And they didn't just look at lab mice, they actually started with human blood plasma.
They did. They compared plasma from NKTCL patients who achieved complete remission against those who were relapsed or refractory. But they paired that real-world clinical data with incredibly advanced lab data using TMT-based proteomic sequencing on those drug-resistant zombie cells.
Tandem mass tag sequencing, or TMT. For anyone who hasn't dug into proteomics lately, TMT is just brilliant.
It really is an elegant tool.
Yeah, you basically attach these heavy chemical isotope tags to the proteins in your samples. So when you run them through a mass spectrometer, you aren't just seeing what proteins are there.
Right, you get much more than that.
You can precisely weigh and quantify their exact abundance across multiple different samples simultaneously.
Exactly. It provides immense clarity. They were sifting through hundreds of proteins, looking for the needle in the haystack. They mapped, I think it was 506 upregulated proteins and 451 downregulated ones.
Wow, that's a massive data set.
It's huge. But when they cross-referenced that lab data with the human patient plasma, the Venn diagram filtered all of that noise down to just 2 proteins.
Just 2.
Just 2, CCath and LCP2.
And CCath didn't survive the statistical scrutiny, right?
No, it didn't. Its expression levels were just too erratic across the samples.
But LCP2 was consistently, significantly depleted in both the chemo-resistant patients and the lab-grown resistant cells.
Consistently depleted, yes.
Which is fascinating because that means the armor isn't something the cancer built, it's a liability the cancer dropped.
That's a great way to put it.
So what exactly is LCP2?
So LCP2 stands for lymphocyte cytosolic protein 2, though you'll often see it in the literature as SLP76.
Okay, SLP76.
Right. In a healthy T cell, it is a critical signaling adapter protein. Think of it like a communication switchboard for T cell receptors.
A switchboard.
Yeah. When the outside of the cell detects a threat or a signal, LCP2 physically bridges the gap to the interior machinery. It tells the cell how to mature, react, and ultimately whether to survive or die.
Oh, I see. So when the cell loses its main communication switchboard, it just goes rogue.
Exactly. It loses its regulatory instructions.
And the predictive power of this is what really caught my eye in the paper. The researchers used ROC analysis.
Receiver operating characteristic curves.
Right, which measure the diagnostic ability of a test. They use that to establish a highly specific cutoff value for LCP2 in a patient's plasma.
Yes, an optical density or OD value of 0.918.
0.918.
And that number is profound. Patients whose LCP2 levels fell below that 0.918 threshold had significantly shorter survival times.
Wow.
The researchers basically proved that a simple blood draw could serve as a direct predictor of chemotherapy failure.
But, correlation isn't causation. To prove that dropping LCP2 actually causes the resistance, they had to play God in the lab a little bit, right?
And they did. They had to test it mechanically.
Right. So they used short hairpin RNA, or shLCP2 knockdown, to artificially silence the gene and remove LCP2 from standard NKTCL cells.
And the results were immediate and aggressive. Without LCP2, those standard cells became incredibly resistant to multiple different chemotherapy drugs overnight.
Just from dropping that one protein?
Yes. They began pumping out massive amounts of that toxic SASP soup, and their aging pathways just skyrocketed. They perfectly recreated the therapy-resistant zombie state simply by shutting down that one communication switchboard.
Okay, time for another quick recap for you listening to make sure we're tracking. Bullet 1 for this section, advanced proteomic sequencing isolated LCP2 as the missing biological link in these chemoresistant tumors.
And bullet 2, low levels of LCP2 correlate directly with high cellular senescence and poor patient survival.
And the third bullet, artificially removing this communication switchboard recreates the dangerous zombie state right in the lab.
Which is a huge validation of their theory.
Definitely. So here's a question for you. If you were a doctor and could simply test a patient's plasma for LCP2 to predict if their chemo would fail, how would that alter your treatment strategy from day one? It completely changes the game.
It truly does. It makes treatment proactive instead of reactive.
Absolutely. But this brings up the most complex and honestly most fascinating question of this whole deep dive. How does a missing switchboard actually create a zombie cell?
Right. What is the actual mechanism?
Yeah, what is the physical, mechanical chain reaction here?
Well, this is where we dive into the phosphoproteomics.
Ooh, fancy word.
I know, right? But it just means we aren't just looking at what proteins exist, we are tracking how they physically interact and modify each other in real time.
All right.
The researchers traced a highly specific molecular algorithm known as the IQG-AP2 LaminA SUV39H1 axis.
Okay, that is a mouthful. Let me try to map this out visually for us.
Go for it.
It's almost like a cellular security system failing. Let's say LCP2, our switchboard, is a security guard, and he's tightly holding the leash of a very aggressive guard dog, which is our next protein, IQGAP2.
That's actually a really great way to picture it. Biologically, LCP2 physically binds to IQGAP2.
Okay.
The researchers confirmed this direct interaction using silver staining and mass spectrometry. So as long as LCP2 is present, IQGAP2 is stabilized.
The guard has a dog.
Right. But when LCP2 vanishes, when our security guard disappears, the levels of IQGAP2 plummet. The leash breaks.
And when that dog gets loose, it trips a silent alarm. In the cell, that silent alarm is a scaffold protein named IQGAP1.
Exactly. The drop in IQGAP2 frees up IQGAP1. Now, this scaffold protein is highly mobile. It rushes away from the cell membrane and travels directly into the nucleus of the cell.
Okay, penetrating the nucleus.
Yes. And once it breaches the nucleus, it targets the nuclear envelope, specifically a structural protein called LaminA.
So the alarm triggers the building's massive steel blast doors to slam shut. Those doors are LaminA.
And slamming shut in this biological context means phosphorylation. IQGAP1 induces phosphorylation of LaminA at two very specific amino acid sites, serine 51 and serine 429.
And what does that do?
Well, phosphorylation fundamentally changes the structural conformation of the protein. When LaminA changes shape, it awakens the final piece of the algorithm, the epigenetic enzyme SUV39H1.
Okay, so the blast doors lock, sealing the evil CEO inside a panic room, and the CEO starts rewriting the rules of the building from the inside. SUV39H1 is the zombie boss.
The zombie boss. I love that. The precise biological term for SUV39H1 is a histone H3K9 methyltransferase.
Let's break that down.
Basically, it alters the architecture of the cell's DNA. DNA is wrapped around these spool-like proteins called histones.
Right.
SUV39H1 adds methyl chemical groups to specific spots on those spools. This causes the DNA to wind up so tightly that the genes responsible for normal growth and cell death are physically hidden. They are silenced.
Wow. So it literally overwrites the genetic accessibility, just locks away the instructions for death.
Exactly. The severe chromatin remodeling drives telomere stress. It physically locks the cell cycle into the G0, G1 phase.
Which means?
The cell cannot divide, but because the apoptotic death pathways are silenced, it absolutely refuses to die.
Okay, so just tracking this cascade. The LCP2 guard vanishes, the IQGP2 dog drops, the IQGB1 alarm rushes the nucleus, the LaminA doors get phosphorylated, and the SUV39H1 boss methylates the histones to lock the cell in stasis.
That's the algorithm.
That chain reaction is the precise mechanical cause of the chemotherapy resistance.
It is a flawless algorithmic survival mechanism.
That is just mind-blowing. Okay, let's lock this in with a quick recap for you listening.
Right.
Bullet 1. The disappearance of LCP2 triggers a precise chain reaction inside the cell.
And bullet 2 is that specific order of molecular dominoes. LCP2 loss leads to lower IQGAP2, which leads to higher IQGAP1, which alters LaminA, which activates SUV39H1.
Perfect. And bullet 3. SUV39H1 acts as the zombie boss, altering DNA to lock the cell into an indestructible dormant state.
Exactly.
So, question for you to ponder. When you picture this molecular relay race, which runner do you think would be the smartest one to target with a new drug to stop the chain reaction entirely?
Oh, that's a very good question to think about.
Because you got to break the chain somewhere. But wait, I'm spotting a missing variable here in this whole setup.
What's that?
The tumor doesn't exist in a vacuum, right? It exists in a living human body. We have an immune system designed to detect and destroy rogue cells. Why are the body's natural T cells just letting this zombie panic room exist?
That is the exact question the researchers asked next.
Oh, really?
Yeah. To figure out what happens in the tumor microenvironment, they designed a co-culture experiment. They took these SUV39H1-driven zombie cancer cells and placed them in a dish with healthy human immune cells, PBMCs or peripheral blood mononuclear cells.
Let me guess. The SAP soup we talked about earlier did some damage.
Damage doesn't quite cover it. The SAP toxins, particularly a cytokine called IL-8, completely brainwash the immune system.
Brainwash them.
Completely. The numbers of actual fighting cells, the CD8 plus effector T cells that secrete interferon gamma to kill cancer, their numbers just plummeted.
The soldiers were wiped out.
Wiped out and replaced by bystanders. The environment saw a massive spike in what oncologists call exhausted T cells.
Exhausted. Like they're just tired.
Biologically tired, yeah. These are immune cells that have been forced to express surface proteins like PD-1, LEG3, and TIM3.
Right, and PD-1 is an immune checkpoint. It's essentially a biological brake pedal.
Exactly.
Normally, it keeps the immune system from attacking healthy tissue, but the cancer coats itself in signals that hit that brake pedal, telling the immune cell, hey, nothing to see here, don't attack.
Precisely. The zombie cells force the immune cells to hit their own brake pedals, and they also saw a massive spike in regulatory T cells, or TREGs, which actively suppress other immune cells.
Wow.
Yeah, the SASP literally exhausts the immune system, turning the body's own defense forces into tired, confused pacifiers.
Okay, but a co-culture in a petri dish is one thing.
Yeah.
Does this complex chain of immune exhaustion and epigenetic lockdown actually happen in a living, breathing body?
It does. And to prove it in vivo, they built a highly specialized animal model. Now, you can't just completely knock out the LCP2 gene in a mouse to test this, because LCP2 is required for fundamental hematopoiesis, the development of blood and immune cells.
Right, so a full knockout would be lethal to the embryo, right?
Exactly, it wouldn't survive. So they used heterozygous LCO2 knockout mice, known as HKO mice.
Heterozygous meaning they only knocked out one copy of the gene.
Exactly, leaving just enough function for the mice to survive. This perfectly simulated a human patient whose LCP2 levels have significantly dropped due to systemic chemotherapy.
Oh, that's clever. So they implant the NKTCL tumors into these HKO mice, hit them with the adriamycin chemo, and the LCP2 drops.
And the tumors became totally resistant.
Just like in the humans.
Exactly. The HKO mice developed massive tumors with intense SA beta-gal blue staining, huge spikes in the aging genes P16 and P21, and severe immunosuppression. The entire algorithm executed perfectly in a living organism.
Unbelievable. Which brings us to the climax of the paper. We know the molecular domino effect. We know the tumor is heavily armored and the immune system is brainwashed. So how do we breach the panic room? What is the solution?
The solution lies in a cutting-edge class of drugs called senolytics.
Senolytic. Seno referring to senescence and aerolytic meaning to destroy.
You got it. These drugs don't try to cure the senescent cells. They are biological assassins designed exclusively to hunt down and clear out aged, dormant cells.
Okay, so what was the specific weapon of choice here? How do you target a cell that's essentially holding its breath?
They used a dual drug combination to attack the mechanism from both ends.
Mark.
The first drug is called Epitalon. Epitalon is a synthetic peptide that works upstream. It modulates telomerase activity and inhibits the broader cellular senescence network that gets triggered the moment LCP2 drops.
So Epitalon cuts the alarm wires before the signal can amplify.
Right.
But that still leaves the CEO locked in the panic room. How did they breach the door to get to SUV39H1?
With the second drug, Chaetocin.
Chaetocin. Okay.
Chaetocin is a highly specific small molecule inhibitor. It physically binds to the active site of SUV39H1, preventing it from transferring those methyl groups to the histone.
So it stops the epigenetic enzyme from winding the DNA shut.
Exactly. It goes straight for the zombie boss.
And when they unleashed this Epitalon and Chaetocin combo on these highly resistant immunosuppressed tumors in the mice, what was the clinical outcome?
Complete reversal.
Really?
Complete. The senolytic combo selectively induced apoptosis in the zombie cells. They essentially forced the invincible cells to finally dismantle themselves. And because the zombie cells were dying off, the toxic SAC cytokine factories shut down. The IL-8 levels plummeted.
And without the toxic brainwashing suit.
The immune system woke up from its coma.
Oh, that's amazing.
The fighting CD8 plus effector T cells rushed right back into the tumor site, while the exhausted PD-1 T cells and suppressive tregs dropped away. By assassinating the zombie cells, they completely reversed the chemoresistance and restored the body's natural ability to fight the tumor.
That is just incredible. Okay, last quick recap for you listening. Bullet 1. Senescent zombie cells secrete toxins that exhaust the immune system, preventing it from fighting the tumor.
Bullet 2. Researchers used a specialized genetic mouse model, the HKO mice, to prove this resistance mechanism happens in living bodies, not just in a dish.
And bullet 3. A combination of 2 senolytic drugs, Epitalon and Chaetocin, successfully assassinated the zombie cells, reversed the resistance, and revived the immune system.
A total victory for that drug combo.
Which begs the question for you. Could this senolytic approach, actively hunting and clearing out aged, dormant cells, be the master key to unlocking treatments for other drug-resistant diseases?
It's a very promising avenue.
It really is. So if we pull back and synthesize what this means for a listener who might be in the medical field or just fascinated by the future of oncology, this research proves that drug resistance in NKTCL is not just about cancer cells randomly mutating to outsmart a chemical.
No, it is far more deliberate and algorithmic than that. These cells are utilizing an ancient biological mechanism. They are intentionally aging themselves into a state of suspended animation.
Wow.
They endure the toxic environment, protect themselves with this LCP2-driven epigenetic armor, and patiently wait to rebuild the tumor stronger later using our own immune system as a shield.
It goes all the way back to our fire and water analogy from the start.
Yeah.
The chemotherapy water didn't just fail to put out the fire, it forced the fire to change its fundamental state of matter.
Exactly.
But now, by understanding this exact chain reaction from LTP2 down to SUV39H1, and by deploying senolytics like Epitalon and Chaetocin, we finally have a highly targeted tool to extinguish it.
It opens up an entirely new therapeutic frontier. If we test a patient's blood, see their LCT2 levels are low, and preemptively use senolytics alongside standard chemo, we could theoretically prevent relapsed and refractory disease before it even starts.
That would be revolutionary. Which actually leaves me with one final, very provocative thought for you to explore on your own. We've just spent this entire deep dive talking about how chemotherapy forces cells into a senescent, aged state to survive, and how we now have senolytic drugs that can hunt down and clear them out.
I know exactly where you're going with this.
Think about the broader implications here. If these drugs safely eliminate aged zombie cells in a tumor, could we eventually use these exact same senolytic therapies, like Epitalon and Chaetocin, not just to cure resistant cancers, but to routinely clear out the natural buildup of senescent cells that cause human aging itself?
The anti-aging angle.
Yeah. Could the cure for cancer resistance also be the fundamental biological master key to the fountain of youth? Something to mull over until our next deep dive?